So yeah, we're going to move on to the first trainee talk of the day. So the trainee talk is by Elizaveta Lavrova, who is a PhD candidate working at the Giga Cyclotron Research Center associated with University of Liège.
Yeah, hello. Thank you for the kind introduction. I'm Elisa Lavrova, and indeed, I'm a PhD candidate in Liège and Maastricht University. And today I would like to talk about a topic which is a little bit less fundamental. So it's related to synthetic data generation from conventional and qMRI data. And here we're moving forward. Yeah. So how we came to the idea of this project. So we have some previous study where we analyzed features extracted from different tissues of MS patients from the qMRI data of these patients. And then we analyzed these features and then performed some classification. But the thing is that to perform the segmentation of white matter hyperintensities for these patients, we needed FLAIR data, just because this is a common MRI modality that is essential for both manual and automated segmentation, and which brings us to the aim of this study. So we noticed that there is something controversial in here, because on one hand, we are performing some really advanced analysis of qMRI data, but on another hand, we need some data that is like conventional MRI, and what if we don't have it in some data sets, or we have retrospective studies, or the data is missing. So in these cases, the state-of-the-art solution is implementation of some deep learning methods for data synthesis, such as GANs. So something like this, when we have one image and then we perform some style transfer to this image, and then, for example, from a summer landscape, we can get something like winter landscape. And we can perform the same thing with brain MR data, so we can just do this cross-modality transfer. But the challenge in here is that if we know that there is some pathological information which is not visible or not very bright on original, on source scan, then it might not be transmitted accurately to the target domain scan. So therefore, within this project, we hypothesized that we can generate FLAIR scans for multiple sclerosis patients with cycle GANs if we use qMRI maps. And in this case, we will not lose any diagnostic information. And this is the data set we were working with. So this is a data set containing qMRI and conventional MRI data of 36 MS patients acquired in Liège in Belgium. And here are the qMRI maps available for this patient. So qMRI map, 4 qMRI maps. And as conventional MRI, we had T1 weighted data, which is basically like T1 weighted first echo that was a part of MPM acquisition protocol. And also we had FLAIR data for these patients, which was acquired like separately, but it was co-registered with SPM. And then qMRI data and conventional T1 weighted data, we consider them as source domain data, and FLAIR data is a target domain data. So it's something we want to synthesize. And how do we do this? We implement cycle GANs, so it's a deep learning method. And for example, we have some input scan of a domain A, so of a modality A, let's say, something like this. And then we implement a generator, so it's a neural net that is trained to perform transfer of a scan from a domain A to a domain B. So basically we have one scan as an input and another scan as an output. And then we have synthetic image, so it's the same patient, but it's a synthetic scan of modality, let's say B for this patient. And then we implement another neural net that is trained to generate image from modality A, of the modality A from the modality B. So basically we make like the back transfer. So from the synthetic scan, we generate another synthetic scan that belongs to the original domain. And basically the data makes a cycle in here. That's why it's called cycle GAN. And in this case, how do we actually distinguish, how we understand that these generators are working properly? So we train 2 more neural nets that are called discriminators. So they have a scan as an input and then just a number as an output. And then this discriminator is aimed to distinguish, like to evaluate if this scan really belongs to some certain domain. And we assume that the generator is trained properly if discriminator cannot distinguish between the synthetic image and the original image. And this is the way we train these 4 neural nets. So we have one loop that goes like from the domain A to B to A. And then we make like the same procedure, but then we go from domain B to domain A and then to domain B. And in this case, all 4 models should be trained properly. And then we are moving to the study pipeline. So as I said, we had some data, so source domain data and target domain data. Then we train cycle GAN model to synthesize FLAIR data, like separately from FLAIR and from qMRI. And then we have some synthetic scans. And of course, we need to evaluate if those are good or bad, if they look like original FLAIR or not. So for this purpose, we perform image comparison. So we compare the original and synthetic scans like at the image level, but it's not the final goal. We are not doing it because we just want to generate the images. So there is some clinical purpose why we want to obtain this FLAIR data. And this is white matter hyperintensity segmentation. So that's why we segment lesions for these MS patients with LST toolbox from both regional and synthetic FLAIR data. And then we also compare the segmentations. And this is the study pipeline. And now we're moving to the results. So first I wanted to show how the training process goes. So as I said, we train data from domain A to B to A, and then from B to A to B. And as a first column, you can see the input, the original data, and then synthetic data, and then so-called cyclic data. So it's a synthetic data that is generated from synthetic data. And we can see that when we train during the first epochs, these scans, they do not really look like scans, these images. And then when we have a look at the data in the last epochs, we can see that indeed synthetic FLAIR data really preserves all the anatomical structures and even white matter hyperintensities in here. And if we have a look even at cyclic data, well, it really looks like real data, maybe a little bit blurry, but still it looks fine. And why the qMRI data is that colorful? It's just because I compose different qMRI maps into RGB channels, that's it. And then we're moving to the numerical estimation of the results. So in here, you can see some examples of the generated data. So first of all, you can see the source T1 weighted data, and then FLAIR data that is obtained from the T1 weighted data. And yeah, it has some anatomical structures and even white matter hyperintensities in here. But then the texture of white matter is a little bit odd, but still it's possible to work with it somehow, maybe. And then there is target FLAIR data just for comparison. And if we move from right to left, like in this direction, then we can see the original qMRI data, again, that is represented in multiple channels. And then the synthetic FLAIR data, which looks much better than synthetic FLAIR data obtained from T1 weighted data. So it has these anatomical structures, white matter hyperintensities, which are much brighter, and also the texture which looks maybe a little bit like not perfect, but still much smoother than in the first case. And here are the numerical results. So in here, the first 2 columns are representing structural similarity and feature-based similarity indices. So these metrics we used in order to compare the images, like synthetic images and the original images on the image level. And the last 2 scores, they obtained for lesion load, and they are obtained when we compared the masks. So something we obtained with LST toolbox. So those are root mean squared error while we calculated lesion load, and also interclass correlation coefficient when we compare the lesion loads in here. And expectedly, if we generate data from qMRI, if we generate FLAIR from qMRI data, we obtain better results compared to if we generate FLAIR data from T1 weighted data, just because we have more channels, more information. It's more descriptive, obviously. So all the scores are higher, like image related are higher, error should be lower, and again, ICC should be higher. These scores are maybe not perfect, but still for the size of a data set, they are quite okay. And what is really optimistic in here is the concordance in lesion load, so 0.0 almost 8 is quite a high score. And this brings us to the conclusion of this project, of this study. So first of all, cycle GAN is a suitable technique for cross-modality synthesis, but if we want to preserve some pathological information, it's better to use qMRI data, if possible, of course. And anyway, it's a pilot project. That's why this technique should be validated on larger data sets and different pathologies and the other MR sequences as well. And talking about the potential application, well, this technique can be applied in retrospective studies when we don't have some sequences, or just for cost reduction if we don't want to have them, and also for data augmentation or imputation purposes. With this, I would like to thank you for your attention, and I would be glad to answer your questions if you have any.
Thanks a lot for your talk. Very interesting. Aurelia, are there any questions in the chat?
Yes, actually, we just received them. So from Christopher Rode, he was asking how many training data sets are needed to get reasonable synthetic data for T1 weighted versus qMRI data?
For the backbone model, we performed training with both 250 epochs, and also if we are working with this cycle GANs, let's say, then it's important to have some epochs with the same learning rate, and then we reduce learning rate until we reach 0. So this particular data set, it contained 36 patients, but the training and validation was performed on just on 24 patients, and still we have this result. So if we are going for higher correspondence on the image level, then I would say that like for positive results, also it depends on the diversity of data, of course, but I would say that it's possible to do it even with less than 100 patients, because even like if we have qMRI data, because even at this point it was okay. But if we're talking about T1 weighted data, I'm a little bit afraid that even if we increase number of epochs, then we might not really approach the good metrics, the metrics we want, just because the data maybe does not represent enough information for this sort of task. But yeah, I would say that maybe it's also hard to say, because on another hand, qMRI data contains more channels, and that's why more channels you have, more subjects you need. That's why I would say that it's a trade-off.
Thank you. So we have another question from Dr. Sridhar Narayanan. Very nice work. A good potential application will be at 70, where the coverage of FLAIR is not great. Have you tried generating FLAIR images from MP2H plus multi-echo gradient echo at 70? This could be interesting for future work.
Yeah, thank you for this question. It would be really interesting, but unfortunately, I didn't have access for such data. That's why I didn't try to do this. But I would say that it's also possible, and it would be interesting to perform the same methodology in order to create high resolution data, because there are some applications where people are trying to increase resolution and they are trying to synthesize the data that is like 7 Tesla-like, but I'm not sure if this synthetic data would contain enough this miniature diagnostic information. But that's a good question, so it would be nice to try, of course.
Thank you. So that's it for now. I don't have any more questions in the chat.
Okay,
Thank
so
you.
I think, thank you. Thanks a lot.